Our first student is Hiba Kamel. She's a PhD candidate in plant science, and she will be talking about trafficking jam in plant cells. With that, the floor is yours.
Thank you, Andrew, and hello everyone and welcome to this presentation. So today I will be discussing the role of pectin secretion in pollen tube growth and the technology that I will be manipulating to gain a greater understanding of pollen tube growth dynamics. So what you might have learned in high school biology is that reproduction of plants happens when a pollen grain, which contains the sperm, lands on the stigma head of a flower and then makes its way all the way to the female gametophyte as such to produce a seed. But that is not how it actually happens. Plant reproduction does begin when the pollen grain lands on the stigma head of a flower. However, what happens next is that it rehydrates and then a tube-like protrusion starts emanating out of the pollen grain. And this tube extends all the way to the female gametophyte, which is contained within the ovule. And then the pollen tube ruptures and the sperm cells are released and they are free to fertilize the female gametophyte. And this is what gives rise to a seed. So essentially, the pollen tube is the conduit for the sperm cells. So here's an image of an actual pollen grain. It has been germinated in vitro, and we're looking at it through a microscope. So this is the pollen tube. Growing this tube is considered the most rapid cell growth process in the plant kingdom. And I just wanted you to note that the actual growth happens from the tip region. So unlike our hair, which grow from the root, pollen tubes actually grow from the tip. So how can pollen tube grow so fast and produce pollen tubes that are thousands of times the length of the initial diameter of the grain? So this question involves a number of factors. So to better answer that, it would be better to look at a diagram. So I'm going to show you that same image again that I have rotated and merge it with a diagram of a pollen tube. So going back to our question, how do these pollen tubes grow? So one contender to answer that is that the pollen tube takes advantage of differential cell wall material properties to extend locally and exclusively at the tip. So what you are seeing here again is a pollen tube, and I want you to notice that it's outlined in a gradient of color going from light green to darker green. So this outlining depicts the cell wall of the pollen tube. And we will not go into the molecules depicted in these boxes, but what we will see is that different molecules are secreted into the cell wall, and it will give either a stretchy material property to the pollen tube or a more stiff or rigid cell wall property, so that we have growth only in one region. So this is some of the components. We have pectin 1 and pectin 2, that these are not their original name, but for simplification, that's what I'm calling them. So pectin 1 is found exclusively at the tip, and it's associated with a more extensible cell wall state. And pectin 2 is more at the shank, and it's associated with a more rigid cell wall state. And then we have protein 1, which turns pectin 1 into pectin 2, but it only does that in this region and not that region. And why? It's because protein 2 inhibits protein 1. All of this together, you do not have to understand the detail, but all of these elements that are secreted into the cell wall all work together to make the cell wall stretchy at the tip and more rigid at the base or the shank. So these components are actually produced inside the cell. The pollen tube is a cell. The pollen grain itself is a cell, and the tube is a part of it. So these components get made, and they need to be carried to the cell wall. So how do they get carried? They get carried through these membrane vesicles, depicted here in these green bubbles. So these membrane vesicles is what I essentially study. So let's take a look at a journey of a membrane vesicle. Membrane vesicles comes from the periphery and then gets secreted in this region. Sometimes it fails to secrete, and instead it accumulates in this, the membrane vesicles accumulate in this very characteristic triangular shape at the tip. Now let's take one of these little guys, these green bubbles, and let's zoom in on what's inside. You might have guessed because I've mentioned it already, but what's inside are these components that I was talking about. So if we zoom in on this little guy, we see a membrane vesicle, and inside are cargoes. We call them cargoes because they're being transported. And these cargoes could be things that I've mentioned already: pectin, protein 1, protein 2. My goal is to actually visualize these cargoes. Why? I want to see how they are sorted and how they are organized, and I want to do that in a conclusive manner. So this is the microscope that is most commonly used to observe these constituents. And with this, you can't actually see membrane vesicles, let alone their content. So to conclusively observe membrane vesicles and their cargo, this is the big guy I'm going to be using. It's called TEM, for transmission electron microscopy. And this microscope, as you can see, is much bigger, much more sophisticated, and it allows us to see things in much more details. Why? Let's look at the resolution limit. So this guy's resolution limit is 180 nanometers, whereas this one's resolution limit is 0.2 nanometers. All that means is that the smallest distance it can observe is 0.2 nanometers, and that's extremely small. It can resolve extremely small, or at least way smaller than this other guy. My membrane vesicles are about 60 nanometers. So they are smaller than the smallest thing this guy can resolve, but bigger than the smallest thing this guy can resolve. So I'm picking this microscope. Let me give you a quick rundown of how I need to prepare my samples to visualize them in the TEM. So this is how it goes. You start with your biological sample. In my case, it's a pollen grain with a germinated pollen tube. And then you need to fix this sample. You immortalize it in that moment in time. How you do that? You either flash freeze it or you chemically fix it with chemicals. And then eventually what you do is you embed your sample in resin, a very hard block. It's actually going to be very tiny. And then what you do is you cut ultra-thin sections of that sample. And you do that with a very sharp diamond knife. And then what you do with those little sections is that you immunolabel them. And how that works is no different than how your body fights off some viruses. It makes antibodies to target them. So I will have antibodies that target my molecules of interest, and that antibody is going to target them. And then another secondary antibody comes on top of that, targeting my primary antibody, and that one will carry gold. So that when I visualize them on the microscope and I bombard them with electron, I will be able to see a dot, a black dot, where the gold particle is. And to just give you an idea of what that will look like, if I cut a very ultra-thin section of this pollen tube, what it will give rise to is an image like this. You see little dots of particles. These are what in this research they were labeling for a specific molecule. What you see here is actually a membrane vesicle, and you see inside it that little molecule that they wanted to see inside it. So in most studies, these important cell membrane constituent that I described as cargos are actually observed at a lower resolution, and this may lead to inconclusive interpretation on how they're secreted, where they are in the cell. So using the big guy microscope, I'll be able to conclusively identify the exact cargo composition of membrane vesicles at the tip of the pollen, and it should look something like this. And this is a simulation of what it would look like. So finally, my research wants to simply validate the sorting of cargos inside membrane vesicles in the tip of pollen tubes. So thank you so much for your attention. And I wanted to thank my supervisor and my coworkers and colleagues. Thank you.